Hello friends, today we are going to discuss what to do once a gene has been cloned. In my last video, I discussed the process of gene cloning in detail, and what happens during that process. We generally isolate the gene of interest, we join that gene of interest into a vector molecule like plasmid. What we get a recombinant DNA molecule. That recombinant DNA molecule is further introduced into a living system that is called a host. What we do next, we culture those transformed host cells, leading to their multiplication, and inside those host cells the rDNA also gets multiplied, and this entire process happens inside controlled lab conditions with the advent of certain enzymes. So what is the product of this gene cloning? It's basically a large population of host cells which are having a large population of recombinant DNA, and inside those recombinant DNA molecules there are multiple copies of our gene of interest. So the question is how we can use those multiple copies of the gene which are lying inside those host cells. One of the most important idea of utilization of those gene of interest is that what we can do, we can induce the expression of those genes inside the host cells itself so as to recover the protein produced from these gene of interest. So the primary way of utilizing the gene of interest is to recover the protein product. You know very well that gene is basically a short part of DNA which contains the genetic information necessary for the formation of a protein, and the protein is able to do its required function. For formation of protein from a given gene, there is a long journey, there is a step-by-step journey, and that entire journey is known as the gene expression. So the very first step of gene expression is basically known as transcription, which is the formation of mRNA by utilizing the information given in a gene. The mRNA is modified post-transcriptionally to give rise to mature mRNA. The mature mRNA is translated and forms the protein, and the protein is modified post-translationally, leading to the formation of the mature protein, and that mature protein starts its function inside cell. So this entire process is known as gene expression. One more thing should be kept in mind, that for this entire process there are three important signals, three important elements which must be present along with the gene so as to facilitate its successful expression. The very first expression signal is promoter, the second is operator, and the third one is ribosome binding site. The promoter is basically a region which is necessary for initiation of the process of transcription. The operator is basically a region which is present downstream to the promoter and which is basically concerned with the control of the transcription level of a particular gene. The ribosome binding site, as the name suggests, it is a site where ribosome binds and initiates the process of translation. So all the three control elements are very critical for the successful carry out of the process of gene expression. So if you want that your gene is expressed here, what you have to ensure, you have to incorporate all the three expression signals into your vector. During cloning, what you have to do, you have to incorporate the gene into the vector Along with the gene, you have to incorporate all the 3 appropriate expression signals so that the gene can be expressed. And one more thing should be kept in mind, that these 3 expression signals must be selected in such a way that they are functional in the selected host. If you are using prokaryote as a host, you should use the expression signals according to a prokaryotic organism. And if you are using a eukaryotic host, you have to choose the expression signals accordingly. Why? Because the expression signals of prokaryote do not work in a eukaryote, and expression signals of a eukaryote do not work in a prokaryote. That's why the most important things are, one is the expression signals are important, and second one is the expression signals should be such that they are compatible with the host in which you are going to clone and express your gene of interest. So if you incorporate the expression signals inside your vector and you grow your transformed host cells in culture, what will happen? The rDNA will replicate itself, leading to the multiplication of the gene of interest, as well as, because you have incorporated the expression signals inside vector itself, the gene will also express itself successfully to make the required protein. Either the protein will remain inside the cells, or the cells may also secrete that protein outside them. In that case, the protein will be lying inside the growth medium. So your protein may be intracellular or your protein may be extracellular, depending upon the pattern of the production of the protein. So what you have to do now, you have to purify your protein from the place where it is found. If it is found inside cells, if it is intracellular, you have to break the cells and purify the protein. And if the cells have released the protein in the medium, if it is extracellular, you have to purify the protein from the medium itself. So at the end of the purification, what you will get your protein. And because that protein has been produced by the advent of recombinant DNA technology, that protein is known as the recombinant protein. The basic concept is, when you are growing your cells through the process of gene cloning, what is being happened here, the cells are dividing by binary fission. If there are 2 cells, they will divide and they will convert into 4 cells. 4 cells will again divide and they will convert into 8 cells. So because the number of cells is increasing exponentially, the number of rDNA will also increase inside cells. And if the number of rDNA is being increased inside cells, number of copies of the gene of interest also being increased inside cells. And as a result of this, the expression levels of those genes are also being increased. So the amount of protein will keep on increasing. It means more you grow your cells, more the amount of protein you get. So this process is very beneficial. You can get enhanced amount of your protein. What you have to do, you have to just keep on culturing the concerned host cells, and you have to purify the protein, whether from inside the host cells or from the growth medium, depending upon the location of the protein. So now let's take an example of such a strategy which happened in 1978. What was that? It was production of human insulin using Escherichia coli as host. what scientists did, they isolated the gene coding for insulin from human cell. Then they introduced that gene into a vector, into a plasmid, and they also incorporated the appropriate expression signals, that is promoter, operator, and ribosome binding site inside the vector. And by joining the gene with the plasmid, they got recombinant DNA. Then they transferred that recombinant DNA into a bacterium named as Escherichia coli that served as a host for the cloning as well as the expression. What they get? They get transformed Escherichia coli cells. What they did next? They did culture of those cells in vitro, and as a result of culture, the cells number got increased, leading to the cloning of the gene of interest as well as the expression of the gene, because they have already incorporated the expression signals in the vector. That's why what they do purification of the insulin. And because this insulin was produced by the advent of recombinant DNA technology, that's why it came to be known as recombinant human insulin, or more appropriately, humulin. So that was the story behind the production of this human insulin. Some facts about this humulin. One is, in 1978, the recombinant human insulin was prepared by David Godel and his colleagues, and they were working in a biotechnology company named as Genentech, and they utilized Escherichia coli for achieving that objective. Thereafter, Genentech and Eli Lilly is an American pharmaceutical company. That was a developer, Genentech, and Genentech along with Eli Lilly, they both signed an agreement to commercialize recombinant human insulin. In October 1982, United States Food and Drug Administration, that is FDA, approved Eli Lilly and Company's humulin. And this humulin was the first human insulin for diabetics created using recombinant DNA technology. It was developed by Genentech in 1978, and it was licensed as well as marketed by Eli Lilly in 1982. And this humulin was the first licensed drug produced using recombinant DNA technology. So these were some of the facts regarding humulin. Now I think that you have some idea in your mind about how we can use the gene of interest that has been cloned inside host to recover the protein product coded by that. Carefully watch the video and post your queries. Okay, that's all for today. Thanks for watching.